Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on Earth, and that's biology. So thank you so much for joining my session this evening. So I hope you guys had a very fabulous weekend and you guys rested well, you guys ate good food, and today we're going to talk about the last lecture of your photosynthesis. So that is, we are just left with almost a concept that is only for 10 minutes, I would say. So we are just left to talk about the CAM plants. So we have to talk about the CAM plants and the factors affecting photosynthesis. Hi, my students classes. So nice, ma'am. Okay, so who are you? Yes, welcome to my session. Hi, Aman. Yes, hello. Very happy good evening. So I hope you're doing well. So thank you so much for joining my session this evening. So please don't forget to like, share, and subscribe. Okay, so in the last class we were talking about what is C2 cycle. So I finished off C2 cycle. I told you how C2 cycle happens. So today we'll be talking about what is CAM plants and what are the factors that affect your photosynthesis. Alright, so it's just for 10 minutes, I would say. We are almost done with all the topics of photosynthesis, and I hope you guys have understood photosynthesis really well. Right, so from Wednesday I'll be starting with biomolecules, so please don't forget to join me on Wednesday at 4:30. Yeah, so we have just 2 topics left to cover. So law of limiting, okay, so this is law of limiting factors. So we just have 3 small concepts, I would say. Okay, so giving you a recap as to how the C2 cycle happens. So C2 cycle happens only in your C3 plants, not in your C4 plants. So we are talking about the C2 cycle. So when carbon dioxide acceptor is RuBP, so here the enzyme is Rubisco, right? So full form of Rubisco is rubulose bisphosphate, carboxylase and oxygenase. Meaning it can bind both to carbon dioxide as well as oxygen. Okay, so Rubisco binds to carbon dioxide under the condition that the concentration of carbon dioxide and oxygen are equal to each other. When the concentration of carbon dioxide and oxygen is equal, carbon dioxide binds to Rubisco. When this concentration is not equal, so Rubisco binds with your oxygen. In that case, you will not have a stable 3 carbon compound, but instead a 2 carbon compound. So when RuBP binds with oxygen, you have 2 carbon phosphoglycolate plus your PGA, which is your 3 carbon compound, but this takes up all your energy, but this does not produce any sugar. So you have no sugar production nor no ATP production, but just ATP is being utilized. So this is in a way a waste reaction, you can say. Alright, so this is your C2 cycle. So C2 cycle first happens in your chloroplast, then peroxisome, and then in your mitochondria. So this mostly happens in your C3 cycle, where the primary carbon dioxide acceptor is your RuBP, which is a 5 carbon compound. Alright, this does not happen in a C4 plant because C4 plants have the intracellular carbon dioxide that is getting produced. So I hope you remember the intracellular carbon dioxide that is getting produced. Yes? Okay, so it is like this. So you have your PEP in your C4 plants plus carbon dioxide. So this is a 3 carbon compound. So here the enzyme is peptidase, and it gets converted into your 4 carbon compound, that is your oxaloacetic acid. So this happens in your mesophyll cells. So your oxaloacetic acid is reversibly converted into malic acid, which is your 4 carbon compound. This actually moves inside your bundle sheath cells. So this malic acid undergoes decarboxylation reaction, where 1 carbon dioxide is released out So plus you have the phosphoglyceric acid, which is your 3-carbon compound. So this carbon dioxide undergoes C3 cycle. So if you see in a C4 plant, your internal carbon dioxide concentration is very high. So it does not have to depend on the atmosphere for more amount of carbon dioxide, right? So that is the main reason why your C4 plant is more efficient than the C3 plant. So a quick glance, your C4 plant have what is called as Kranz anatomy, which ensures it has more chloroplast and it has more biomass when it comes to photosynthesis. Alright, yes? Fine. So let's move further to CAM pathway. So what is this CAM pathway? So you would have come across, so C3, C4, C2 and CAM pathway. So these are pretty common. So the CAM stands for cras-leucan acid metabolism. So crassulacean acid metabolism. So that is how you do it. So here carbon dioxide fixation happens during the night. So in C3 and C4 plant, the fixation happens during the day, whereas here it happens during the night. Alright, the stomata remain closed during the day to reduce transpiration, because when your stomata remains open during the day, you have loss of water happening, that is transpiration. So these plants are very smart that they do not open their stomata during the day and they open it only during the night. Right, so such a stomata which opens during the night, it is called as the stochoactive stomata, or the stochoactive opening, meaning which opens only during the night, right? So oxaloacetic acid is formed due to carboxylation as in C4 plants. So your CAM plants have the same pathway as your C4 plants, but the only change happens during the night. The stomata opens during the night and closes during the day. So that is all the difference that it has. So it is important for the survival of succulents, that is, I would say, xerophiles. The plants that live in very dry conditions, example your desert-like conditions, right? So they have to conserve water at all costs. So they have to reduce transpiration. So thus the stomata open during the night. Alright, so with this your CAM pathway is done. So I could show you a cycle. So they typically follow your C4 cycle. So the primary carbon dioxide acceptor is your PEP, and they form your first stable compound is your oxaloacetic acid. This is your 4-carbon compound. Everything is the same, but the only difference is the stomata opens at night and it closes during the day. That is all the difference it has. Yes, ma'am, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Okay, so let's see. So you have PEP. So at night it takes up the carbon dioxide. Okay, with the carbon dioxide is in the form of bicarbonate ions. So with the help of peptidase, it forms your stable component, that is oxaloacetic acid. So this is converted into your malic acid. So malic acid, when it moves, so all these happens during the day. So it stays in the oxaloacetic acid stage all through the night, and in the morning it gets converted to malic acid. So malic acid gets converted into phosphoglyceraldehyde, and then you have regeneration. So that is the only difference in your CAM plants and in your C4 plants. I hope it was clear. Yes? Any doubt? Okay, so now we are going to talk about, so far we have spoken about C3 cycle. We spoke about C4 cycle, so we spoke about C2, and we spoke about CAM plants, right? So can you quickly tell me who's the acceptor in all these cases? Who's the carbon dioxide acceptor in all these cases? Quickly, let's do a quick recap. So here the carbon dioxide acceptor is RUBP, which is a 5-carbon compound. Here it is PEP, phosphoenol pyruvate, which is a 3-carbon compound. So C2, again here your acceptor is RUBP, which is a 5-carbon compound. Here it is again PEP, which is a 3-carbon compound. So C4 and CAM plants are almost the same, and C2 and C3 cycle are almost the same, right? Okay, so what is the first stable compound? So here the stable compound is glyceraldehyde, phosphoglyceraldehyde, which is your 3-carbon compound. Here it is your oxaloacetic acid, which is your 4-carbon compound. Here it is your phosphoglycerate, which is your 2-carbon compound. And here it is again oxaloacetic acid, which is your 4-carbon compound. So what does unique about C3 plants? It happens in all the plants, all type of plants. So CAM plants, it is special feature or the unique feature is your Kranz anatomy. So this happens when the carbon dioxide concentration is not equal to oxygen concentration. Here the stomata opens during the night. So this is the main difference, alright. So yeah, so with this we are done with all the pathways successfully. So now we are going to talk about factors affecting photosynthesis. So here we go. So rate of photosynthesis is important in determining the yield of plants, including crop plants, right? For example, you are preparing for NEET. So each and every mock test that you write is important in your final destination or for your final exam. Right? So much in the same way, the rate of photosynthesis determines the yield of the plant. So photosynthesis is under the influence of a lot of factors, internal and external. So your preparation for NEET exam is also based on a number of factors, external and internal. So let's say a lot of internal factors play a role. Your health conditions, your interest to study, right? Your dedication, your hard work, all these come from within, right? So that is your internal factor. External factor, disturbance at home, disturbance by friends, your phone is a distraction, your parents sometimes if they start fighting, that is a distraction. All these are external factors, right? So a lot of things affect you when you're preparing for an exam or when you're preparing for your life. So here also for photosynthesis, you have a lot of internal as well as external factors affecting it, okay? So let's see. So plant factors like number, size, age and orientation of leaf. If it's a plant with a large number of leaves, so large chlorophyll, right? Large number of chloroplast. The size, is the leaf large or if it's small. What is the age of the plant? Is it a very old plant or it is just germinating, right? So what is the orientation? How are the leaves arranged? What type of phyllotaxy it has? What type of mesophyll cells and the chloroplast, amount of chloroplast it has, what is the internal carbon dioxide concentration, meaning whether it is C3 or C4, and the amount of chlorophyll that it has. So all these are the plant factors, meaning the factors that are within the plant which affect the process of photosynthesis, alright. So plant or internal factors are dependent on the genetic predisposition and the growth of the plant. So all these are based on the genetics of the plant, right? How much chlorophyll it will have, you and I cannot decide. Only the genes of that plant decides, right? So this is your genetic factor. But you have a number of external factors like the availability of sunlight, the temperature, the carbon dioxide, the concentration of water, etc. Right? So it's carbon dioxide concentration and water. So you have 4 factors. So 1, sunlight, 2, temperature, 3, carbon dioxide concentration and 4, water. So these are by far the most important external factors that affect the process of photosynthesis. At any point, the rate will be determined by the factor that is available at the suboptimum levels, for which we have the law of limiting factors. So let me explain this in a very easy way. Yes, good evening. I am fried chicken. Oh my god. Yes, a very happy good evening. So thank you so much for joining my lecture today, fried chicken. So I guess you're very crazy about chicken. So and please don't forget to like, share and subscribe. Yeah. Okay. So how do I explain Blackman's law of limiting factor? For example, imagine you have 3 subjects. We have 3 subjects in NEET: physics, chemistry and bio. Or you can say physics, chemistry, botany, zoology, or 4 subjects. Okay. If at all you're very weak in physics, that is your success determining subject. So if the other 3 papers go well and if you do physics well, your exam is a big hit. And if your physics spoils, everything else spoils, right? It takes off your happiness. So that is the rate limiting one. So is it for plant. So we spoke about 4 factors which are essential for plant. Say for example, sunlight is a bit lower than the optimal level. So in that case, when sunlight is more, then it is a big hit. If sunlight is less, it is a big flop, right? So that rate determining factor is what decides the rate of the process. So that is what is law of Blackman's limiting factor. So Blackman only postulated this theory, I would say. So if a chemical process is affected by more than one factor, so here the process is photosynthesis, it is affected by 4 factors. So first one is carbon dioxide, water, you have sunlight and temperature, right? Then the rate will be determined by the factor which is nearest to its minimal value, okay, where it is least weak, okay. It is a factor which directly affects the process if its quantity is changed, right? So if it's higher, then it's a big hit. If it is less than the optimum level, it's a flop, right? So that is what determines it. So let's talk about each of the factors. So I will just talk in short about each of the factors, nothing much. So light intensity at which the rate of photosynthesis is equal to the rate of respiration, that is called as compensation point. So in photosynthesis, oxygen is produced, right? So in respiration, oxygen is consumed. So when this is equal, that is called as compensation point. Light intensity, if it increases above the compensation point, rate of photosynthesis increases, right? So if the light is more, then more photosynthesis happens, so it is going beyond. Degradation of the chlorophyll molecule can happen at high light intensity, that is your photooxidation. So you have accessory pigments to prevent your photooxidation. So photooxidation of chlorophyll is under very high light intensity. So that term is called as solarization. So rate of photosynthesis is maximum in the red light, red to the blue light. So where you have the wavelength of your chief chlorophyll pigment, that is chlorophyll A. So the rate of chlorophyll is minimum in the green light. Rate of chlorophyll photosynthesis is more in the intermittent light than in the continuous light, right? So if you have exams continuously, you don't perform well. So if you have once in a while exam, yes, you have the seriousness to do, right? So is plant. So continuously, if it has a lot of light, so that will make the plants lose a lot of water, right? So intermittent light is what is actually very good for your plant. So let's see how carbon dioxide affects it. So carbon dioxide concentration, when it is increased from 0.03% to 1%, this is less, this is high, the rate of photosynthesis increases. More carbon dioxide, you have more fixation, right? So carbon dioxide, if it is more than 1%, then the rate of photosynthesis decreases because you have to suck in a lot of water. So that leads to transpiration, right? So it is a common limiting factor. Rate of photosynthesis decrease in high concentration of oxygen because it leads to your C4-C2 cycle. So there's something called as Warburg effect, the inhibitory effect of carbon oxygen on your photosynthetic rate. So when your oxygen concentration is more than the carbon dioxide, so this binds to your RUBP, thus forming the C2 cycle. Right? So this is your Warburg effect, the inhibitory effect of oxygen on the photosynthetic rate, right? Yeah, this is about your photosynthesis. Sorry, about your carbon dioxide. So with this, we come to the end of your chapter of photosynthesis in higher plants. So I will give you time till Friday. So Friday, we are going to play a Menti on the same. Alright? Yes? Is that fine? So Friday we'll have a Menti in the same chapter of photosynthesis. So Wednesday I'll be starting with biomolecules because I want you to give enough time for your preparation. Alright? Yes? So thank you so much for watching my lecture, and if you genuinely liked my way of teaching, so please do like, share and subscribe. And thank you so much for watching my lecture. So bye and stay tuned till Wednesday. Thank you.